Hello. We have discussed about the polymerization of DNA. We have discussed about this several enzymes that are involved in DNA polymerization. There are yet many other proteins that are required for polymerization to occur. I am Dr. BCMR, Assistant Professor at Shastra. We will discuss a few more things about the replication fork and how it functions. So the important aspect of polymerization we have seen is that it happens always from 5' to 3' direction. That means the 5', the base that it has, would have had a 3' OH to which more number of bases are added. That is how polymerization occurs or happens. If we want to remember it, it's easier to put it as almost all template reading happens from 5' to 3' direction. DNA polymerizes 5' to 3' direction. RNA polymerizes 5' to 3' direction. And even ribosome, when it is reading the mRNA, will read from 5' to 3'. Somehow evolution has stuck on to that 5' to 3'. There are issues as we discussed with DNA polymerase. DNA polymerase polymerizes 5' to 3' direction, but one of the problems is that it cannot initiate replication by itself. So that is a big problem. That means some primer should have been there. In this case, the primer is RNA molecule. So if we have a primer here, it would have been 5' here. Primer is a small RNA molecule that is synthesized by primase, an enzyme, which is a primase is nothing but a dedicated RNA polymerase, the RNA polymerase that is dedicated for use in replication. RNA polymerase, on the contrary to DNA polymerase, can initiate polymerization by itself. It doesn't depend on 3' OH. Like we discussed in DNA polymerase needs a primer template junction. The primer is important. It can only add more bases to the primer or the 3' OH. So we have a small primer synthesized that is shown in green here, and it falls off and then DNA polymerase takes over and synthesizes the DNA in this direction. And if we notice, the direction of the polymerization is from 5' to 3'. This is the 3' side, right? And if we look at the bottom strand, the strand here, it would require a primer, and that primer would be something like this. And because polymerization happens only 5' to 3' direction, and so we'll have to use it that way. And then this is the 3' OH, and then a new DNA strand would be synthesized this way, 5' to 3'. And what happens next is the helicase would have opened up more DNA. The hydrogen bonds, this is the unreplicated region, and now this is the replicated region. So helicase would disrupt more of these hydrogen bonds here and assume that this is the outstretched one. So I would just put the primers and other details that were already given. I would put a primer here, and there is a primer here from the previous step, and then we have the new DNA synthesized on both the cases, something like this and something like this. And we have more DNA that is opened up by helicase. And now we have a 3' OH here. So DNA polymerase would just continue to synthesize this way, in this direction, something like that. And whereas the other one, there is nothing more to synthesize here, and there is single-stranded DNA here which should be synthesized, and the DNA polymerase cannot initiate synthesis by itself. So again, a new RNA primer should have been made, and that is now taken up by the DNA polymerase, and then it makes up another fragment such as this. So I hope we see that one strand is continuously made, which is referred to as the leading strand, and the other strand is made discontinuously, which is referred to as the lagging strand. And on the lagging strand we would find several fragments such as these, and these are referred to as the Okazaki fragments. These are discovered by Okazaki. And so in the next step we will have further opening or denaturation by helicase, and then further synthesis. So the replication fork is easy to understand as long as we follow some of the things. The template that we are drawing should have been marked or labeled with the 5' to 3', the polarity, and we should always remember that polymerization always occurs from 5' to 3', and that DNA polymerase always requires a primer. So if assume that the replication is finished, and then there is still a problem, because we have small stretches of RNA such as the primers here, here and here, and that's not complete DNA. So the RNA should be removed and DNA should be made, and these RNAs are removed by an enzyme called as RNase H. So basically if we understand or see it together, here in this small stretch we have a hybrid of DNA and RNA, right? So there is an enzyme called as RNase H, which would degrade the RNA that is hybridized to DNA. So it would degrade only the RNA that is bound to the DNA. And once that is done, we now have free 3' OH. This would be 3' OH, and that could be taken up by DNA polymerase, and DNA polymerase will fill up these gaps as like this, something like that, or they will fill up these gaps. There is still a small nick that is left. So the DNA polymerase that usually fills up these gaps that are made by elimination of the RNA primer is DNA polymerase 1. DNA polymerase 1 is an enzyme that has low processivity, low rate of polymerization as well, and it is predominantly used in replication, filling up the gaps left out by elimination after the elimination of the RNA primers. And it's also a special one because it has polymerization activity, it has 3' to 5' proofreading exonuclease activity, and it also has 5' to 3' exonuclease activity. It's a kind of all-rounder, but it's not very fast. So we have the problem is that we still have some gaps. That is, we have there are two things. First thing we will discuss is about the DNA polymerase itself. DNA polymerase does not work by itself. It actually is a holoenzyme, which we will discuss later, which forms a complex, and both these strands are simultaneously synthesized. Let's say it here. The strand here is synthesized in this direction, and I hope you see that the replication fork is moving in this direction. It is still here in this figure. More of this DNA is opened up. That means the replication fork was here and it is now has moved in this direction. And let's look at the direction of polymerization. This is the 5' OH. We had a primer here, and then we have 3' OH, 5' phosphate and 3' OH, and then we have continuous synthesis. So for the leading strand, the direction of polymerization is also in the same. It is the same as in the direction of the movement of replication fork. Let's look at the lagging strand. In the lagging strand, there is some big difference, because polymerization was actually happening in this direction, although discontinuously. So the polymerization on the lagging strand is opposite to the direction of the movement of the replication fork. How does nature solve it? Nature is full of these problems, and nature or comes, so new logics will emerge that will allow the existence of the life. And how does it solve such a serious question? It is simple. For life, it is, say, if I use this same place, I'm going to erase this part a little bit and solve the problem of direction. And this problem of direction can be solved it this way. Now the DNA is the strand, the bottom strand is made in this way. And assume that now I will just write down the primes so that it will be easy to access. Say if I had a primer here, something like this, the primer would be 5' and 3'. So while one DNA polymerase is synthesizing this strand, another DNA polymerase is synthesizing this strand. These two are connected at the replication fork. And this DNA polymerase here is synthesizing DNA on this strand like this. Now the direction of replication fork, the direction of the polymerization on the lagging strand is also matching that of the or on the same direction. So that is how nature solves. It twists one of the strands and it polymerizes. And now the DNA polymerase, they both are intact as a holoenzyme, and they are moving towards the replication fork. I mean, in the direction of the replication fork. And that is the reason why it is also discontinuous. Because once this fragment polymerization has occurred to this end, this DNA is released and a new loop is made on which a new Okazaki fragment would be made. The Okazaki fragments could be about or more than 200 bases set as a stretch. And that is why lagging strand has is synthesized discontinuously. And once we have a gap, say this is a DNA molecule, and assume that this is the new one synthesized. There was RNA primer here, assume that, and the RNA primer was removed by RNase H. We now have a gap that is taken up by DNA polymerase, and the DNA polymerase has filled up the gap until there. What is here, if you see it, is a small nick. The phosphodiester, the backbone is not continuous. There is one small cleavage, and that should be filled or connected. Until then, it is not a continuous strand of DNA. It would be considered as a damage. To solve it, we have an enzyme that is called the DNA ligase. DNA ligase is an enzyme that connects two closely related filled. All the bases or nucleotide wise, they are complete, except there is some gap in the phosphate backbone, and that should be connected. The phosphate bond, a diester bond, should be made, and that is catalyzed by DNA ligase. DNA ligase comes in an activated form with AMP. It takes up ATP and forms two inorganic phosphate is released, two of them, and then activated AMP DNA ligase is formed, where the AMP is transferred to the 5' phosphate of the base here, or the nucleotide here, and here we have the 3' OH, which would then get activated and would make an attack on this and form a phosphodiester bond. And that is how the DNA ligase seals all the nicks that are formed during replication or even during repair. I hope we have a better understanding of how things work in replication, and there are yet more things that we need to discuss. We'll discuss in the next video. Thank you very much.